So today we are going to know about the synthetic genomics. It is synthetic built genome whose formation involve either genetic modification on pre-existing life form or artificial gene synthesis to create a new DNA or entire life form. This field that study the synthetic genome, it is known as synthetic genomics. So soon after the discovery of restriction into nuclease and ligase, the field of genetics began using tools to assemble artificial sequence, that too from a small fragment by naturally occurring DNA or synthetic occurring DNA. So advantages is using the recombinant approach as opposed of the continuous DNA synthesis stem from the inverse relationship that exists between the synthetic DNA length and percent purity of that synthetic length. So if you don't got it, it's like synthesizing long sequence. The number of error containing clone increases due to inherent error rate of current technology. Like recombinant DNA technology is more commonly used in construction of fusion proteins and plasmids. So several techniques with large capacities have emerged, allowing the construction of entire genome for making the entire genome. So there is a polymerase cyclic assembly. So what happens in this polymerase cyclic assembly? It is a series of oligonucleotide which is 40 to 60 nucleotide long that together constitute both strand of DNA and they are synthesized. So these oligo are designed in such a way that a single oligo from one strand, its length is approximately 20 nucleotide at each end, and this is complementary to the sequence of different oligos on the opposite strand. So they create a region of overlap. So the entire set is processed through hybridization at 60 degree and elongation by Taq polymerase and a stranded ligase. So denaturation at 95 degrees Celsius, it form a progressively long contiguous strand and ultimately result in final genome. So this PCA was used to generate the first synthetic genome in the history, that of 5X174 virus. Then there is another assembly method, that is the Gibson assembly method. So the blue arrow represent DNA cassettes which could be an 6 kb kilobase each. For each example, the orange segment represent areas of identical DNA sequence. So this process can be carried out with multiple initial cassettes. So the Gibson assembly method was designed by Daniel Gibson during the time at the Winter Institute, required at the set of double-stranded DNA cassette so that constitute the entire genome being synthesized. So they differ uh, from these sequence which contain a homology to other cassette for the purpose of recombination. So polymerase cyclic assembly and Gibson assembly is single step isothermal reaction with large sequence length capacity. So it is used in the place of polymerase cycling assembly for genome larger than 6 kb, where it is used where there is a genome of larger than 6 kb. So the T5 exonuclease perform the Chew-back reaction at the terminal segment. It is working from 5' to 3' direction and produce the complementary overhangs. So overhang hybridize to each other and the fusion of the DNA polymerase fill in any missing nucleotide, and the nicks are sealed with ligase, that is a gap that are sealed with ligase. So the genome capable of being synthesized using this method alone is limited because the DNA increases its length. They require propagation in vitro in order to continue the hybridization according to the Gibbs assembly is often used in conjugation with transformation associated recombination to synthesize the genome, where several hundred kilobases are required in size. So it is not alone which is performing in conjugation. It used the transformation associated recombination. So let's know about the transforming association recombination. So there is a gap repair cloning done where the arrow represent DNA contigs The segments of the same color represent complementary or identical sequence, and there is specialized primer with extension. They are used as a PCR polymerase chain reaction to generate the region of homology at the terminal end of the DNA contigs. So the goal of the transformation association recombination TAR technology in synthetic genome is to combine DNA by the mean of homologous recombination performed by each artificial chromosome, that is a YAC, for important in the CEN element with the YAC vector, so which correspond to the each centromere. So this sequence gives a vector the ability to behave the chromosomal manner, so thereby it allow it to perform the homologous recombination. So DNA cassette is a segment of DNA which often synthesized or amplified by the PCR that can be inserted in a large DNA molecule such as the plasmid or gene to modify its sequence. So it is modular component. It is used in genetic engineering for various purposes including gene editing, protein engineering, and creating library of mutant gene. So DNA cassette is typically a double-stranded DNA fragment with specific sequence. It can be short of 10 base pair or long 100 base pair. So this cassette is synthesized chemically or generated by PCR using a primer with a degraded base for mutagenesis. They are often designed with restriction enzyme recognition site at their end to facilitate the insertion into a target DNA. So this can be used and replace the portion of gene and introduce a new gene or add a specific regular element. There is a DNA cassette crossing over events, region of homology, SYN sequence, and TAR vector. That is a transformation associated recombination crossover event occurring between the region of homologous across the cassette and YAC vector, thereby connecting the small DNA sequence into a one large contig. So the first gap repair cloning is performed to generate the region of homology flanking the DNA contigs and the gap repair cloning is particularly formed the PCR in which the specialized primer with extension beyond the sequence of the DNA target are utilized. So DNA cascade are exposed to the YAC vector and it drive the process of homologous recombination and connect the DNA cascade. So polymerase, cyclic assembly, and TAR technology are used together to construct 600 kilobase mycoplasm genitalium genome in 2008. So the first synthetic organism it was ever created, that is mycoplasm genitalium, which consists of 600 kb. So what was used? Polymerase, cyclic assembly, and TAR technology together they were used for this construction. So similar steps were taken in synthesizing the large mycoplasm mycoid genome a few years later. So general creation of synthetic genome, it is difficult to direct the synthetic oligonucleotide larger than approximately 200 base pairs and maintaining the high fragility. So small oligonucleotide which are around 5 to 20 base pairs, they are combined to create a genomic size oligonucleotide. So in the previous method it was stitching smaller strand involving T4 polynucleotide ligase. So the modern technology use PCR based methods to improve on this method and increasing the speed and fidelity. To increase the fidelity, PCR based method can include an error reversal step in which the nuclease recognize and cut the mismatch base pairs. So it recognizes the possible because error usually cause structural budget and abnormalities in the DNA. So 4 MB E. coli genome created in May 2019 hold the record for largest synthetic genomic size. Thanks for watching. Do like, share and subscribe.